So in the past, when we've talked about photosynthesis, we've talked about quote-unquote regular photosynthesis and how those plants are actually doing something called C3 photosynthesis. And they do this C3 cycle, which is creating a 3-carbon molecule, which is going to store or fix the carbon dioxide. And in this cycle, they use a particular enzyme called Rubisco. Let's quickly draw what that looks like. We need carbon dioxide and we need RUBP. Those two will combine thanks to our enzyme Rubisco. And from there, they will create a 6-carbon molecule. That 6-carbon molecule eventually breaks down into a 3-carbon molecule that we know as PGA. Then that PGA will turn into PGAL. And that PGA can either be used to recycle back and create more RUBP, or we can use some of it to create our glucose. So obviously I don't have all of my numbers right here, but this is a typical C3 cycle. Let's actually just add those numbers in. We need 6 CO2. We're going to need 6 of our RUBPs. We typically are going to create overall 12 PGA and 12 PGAL. 10 of the PGALs are going to be sent back, and 2 PGAL are used to create glucose. But if instead we are going to not do this C3 cycle, because if, for instance, we don't have any CO2 available and only oxygen is available, we might only have that oxygen molecule available to attach to that RUBP instead of CO2. So C4, when we only have that O2, and that's instead of CO2, we call this process photorespiration. When photorespiration happens, let's look at what happens to the actual normal C3 cycle. Photorespiration is not a good thing. So I sort of summarized what normally happens in your C3 cycle, left out a bunch of steps, but these are the important players. So if instead of, and when we don't have this CO2, if instead we only had 6 oxygen, the problem is that we're losing out on the carbon that we usually combine with the RUBP. RUBP has those 5 carbons, now we have 0 carbons to add to it. And when that happens, don't worry again about specifics here, but we're not able to make all 12 of these PGALs. Instead, we're only able to make 10 when we have oxygen available. But the issue with this is if there are only 10 available, we need 10 to regenerate our RUBP. So we're not able to send any PGAL to turn into glucose. So what's shown here in pink is what we call photorespiration. And again, all of these 10 PGALs have to be sent back to regenerate the RUBP. And we're not able to do any of the creation of glucose. We have 0 left to create the glucose. So this process of photorespiration obviously is not a great thing, and every now and then sometimes oxygen will come in instead of CO2, and so how do plants deal with this because they aren't able to make glucose from it? So let's take a look. Here is a leaf, kind of. Let's have some veins on it. Whoop. And then on there we know that leaves need to have stomata. Show that in yellowish. Okay. And we know that the stomata controls what enters and what leaves the leaf. So let's say that this here is our lower epidermis with our guard cells on both sides, and the space between here is the actual stomata, where we're going to have our gases enter and leave, right? We need CO2 to enter and oxygen to leave, but sometimes it's opposite. So I'll just leave it as a plain arrow. But as we know, above that we have our spongy mesophyll, and usually there's space between the actual mesophyll cells. That's why it's considered spongy. And the space between the cells allows it such that air can enter. So you can have your CO2, oxygen, and also nitrogen that can come in. And we need CO2 to be able to do the typical Calvin cycle. And not all plants are going to have adaptations to help them combat this issue of photorespiration. And if you recall, photorespiration is what happens when you have oxygen that is going to try to bind with your RUBP because there is no CO2 available. And when that happens, we can't create any glucose. But the plants that do have adaptations to combat photorespiration are called C4 plants. And what they do is, instead of the typical photosynthesis, they will do C4 photosynthesis. We know regular plants do their C3 respiration, where they make a 3-carbon chain for their G3P or P-gal. But in C4 photosynthesis, this C4 must end up with a 4-carbon molecule. So let's see what that actual cycle looks like instead. We need CO2, and instead of combining with RUBP, it's going to combine with something called PEP, P-E-P. And PEP is a 3-carbon molecule. 1, 2, 3. And together it will combine to create a 4-carbon molecule, which hopefully you know from our group challenge. We call this oxaloacetate. That oxaloacetate can be broken down and we can create another 4-carbon molecule. We call that malate, also sometimes known as aspartate. But I think in your research you all found the term malate instead. And what we can do is we can break off a carbon from the malate when we need it. So we can, yes, we want to get back to our PEP and recycle that, but to get from this original 4-carbon molecule back to this 3-carbon molecule, we'll take one CO2 off to be used. And then that brings us back to the 3-carbon PEP that's recycled, and it can continue to do this cycle. Remember that C3, that cycle is mediated by the enzyme Rubisco, but for the C4 cycle, it is this reaction is facilitated by something called PEP carboxylase. T-A-R-B. This whole thing allows the plant to avoid photorespiration, and it's only going to fix carbon, and oxygen won't be fixed. And that's a good thing, because that would mean it would be photorespiration, and we wouldn't be able to create any P-gal or to turn into glucose. But let's look back here. Why do we take this 4-carbon oxaloacetate and then turn it into malate or aspartate? We actually do the conversion. What's the point if we're only making more carbon dioxide, right? But malate, that is made in the mesophyll. Let's write that in. It can then be transferred to the bundle sheath cells, which we know are in that vascular bundle or in the vein. If this here is our vein, we know around the exterior will be our bundle sheath cells, and they're going to surround these other tube-like things that will transport water and food, right, our xylem and our phloem. And then outside of the bundle sheath cells, as you guys saw in lab, we're going to have these other cells, and these are your mesophyll cells. And the mesophyll cells are storing the malate or the aspartate. Then they'll be close enough to the bundle sheath cells where the normal C4 process happens. So it can just transfer it right in and allow the process to continue. So to review, for C4 plants, they are going to take the carbon dioxide when it's available to them, and they're going to put it into a 4-carbon molecule before it goes into the Calvin cycle. it will store it so that if the stomata is closed and it doesn't have access to it, it can then use it when needed. C4 plants, as you've learned, they're going to keep their stomata closed when it's hot out so that they can conserve water. But even when carbon dioxide is low and the stomata is closed, it can still do this process that we have shown here. And the 4-carbon molecule is going to act as sort of a carbon shuttle. It'll donate the carbon dioxide to the Calvin cycle. Some examples of C4 plants are things like corn, and also things like sugarcane. And often when we've talked about C4 plants, we've also talked about things like cacti and aloe, and those are a type of C4 plants that we actually call CAM plants. And CAM plants, they are actually only going to open their stomata at night to let in carbon dioxide. And this is because they know that they're in a really hot climate, so it needs to conserve as much of its water as possible. And it'll take the carbon dioxide that it gets at night and it will store it in the 4-carbon molecule, oxaloacetate, and bank it at night. And then it will release the carbon dioxide to the Calvin cycle during the day. So hopefully from this screencast you understand this is the typical cycle that happens for C3 plants, but when we get to C4 plants, or when plants don't have readily available CO2, they undergo this process of photorespiration. Photorespiration is a bad thing and plants want to avoid it. So C3 plants, when it's really, really hot out and they're not able to do as much of this, then they're going to go through this. They're not going to be able to do as much photosynthesis, and they'll go through more of this photorespiration. C4 plants, in contrast, they have this process that they can use to store their CO2 so that during the day they can access it when their stomata is closed and carbon dioxide isn't available. They break down the oxaloacetate into malate. From the malate they take 1 carbon dioxide off, then that can be fed into this cycle to be able to create the glucose and pCals. The only difference between C4 and CAM plants has to do with when they are actually storing their CO2 and when their stomata is open, right? So the C4 plant will close its stomata whenever it's hot out, just so it can conserve its water. Doesn't depend on amount of light. Versus the CAM plants, only at night when the sun is down will it open up and allow the CO2 in so that it can store it in the form of malate and oxaloacetate to utilize when photorespiration might be happening. And that's it for this screencast.